tu hebu niambie.
Si ile yako tu hiyo. Unaona hiyo yako umeweka? Unaweza ungeweka hata na nini? Unaweka na namba, hata namba yangu. Hata picha ungeweza weka picha wale watu wawili wa mimi na wewe. Just still tu nataka nitumie hiyo nini?
Haina shida. So huku umesema hivyo. Na mimi nataka hapa ime reflect iko live chatonga. Kwa live zai. Share link, copy link, naenda kushare. Sasa tuko live.
Just kidogo shida ni kitu kidogo. Sasa wewe ndio nitaenda. Nienda.
Good morning everyone. Whoever is in, kindly type in uko ndani ni kuone. Good morning everyone. Whoever is in, kindly type in uko ndani kuone. So we will start our today's lesson. Tutaanza lesson yetu ya leo. I hope you can hear me clearly. Let everyone who is in ensure that you type on the comment section uniambie ni nani yako ndani. Yes. Who is in? Aliye ndani, type there. Tuanze. Tujue nani yako ndani. Esther, Njeri Njogona. Nani mwingine? Naona tuko around 5. Ian, are you in? Ian, uko ndani? Haya, wacha tuanze. The first question. The following diagram shows an apparatus used in ecological studies. It is used in ecological studies. We are going to open this one using Drive. So that we can be able to edit by lighting. The following diagram shows an apparatus used in ecological studies. Name the apparatus. So what is that apparatus? You can type it on the comment section. Remember, we are collaborating. This apparatus is called a putter. It is called a putter. It is called a putter. And the function of the putter, even if we have not been asked to write down the function, it is to suck crawling animals from rock surfaces, and barks of trees. So these are some of the apparatus we use. So you can take note of this one. I want you to give me the function of a sweep net, the bait trap, pitfall trap, and a pair of forceps. These are also other apparatus that are used in ecological studies. So you can kindly also list down their functions, and that will help you to navigate through your personal revision. Let us proceed to question number 2. No, B. Why is glass preferred in making of the part labeled E? Why do we prefer glass? The reason why we prefer glass is because glass is transparent. Glass is transparent. We prefer glass because glass is transparent, therefore allows light. It therefore allows light. Therefore, it allows light into the specimen niche. Into the specimen niche. So that is the role of the glass. It is transparent and therefore allows light through towards the specimen. So tutaendelea. Tirahabi zaman. What do you mean? Tuendelee. Number 2, name the kingdom whose members are all microscopic. So the kingdom whose members are all microscopic is kingdom Monella. Kingdom Monella consists of all bacteria. All bacteria are microscopic. So it entails bacteria. State 2 diseases caused by organisms belonging to the kingdom that we have just mentioned in 2A. So bacterial diseases, some of the bacterial diseases, we have, for example, tuberculosis. We have gonorrhea. We have syphilis. Gonorrhea. So those are some of the, we also have livestock diseases like anthrax, yeah, caused by bacteria. These are some of the diseases caused by bacteria. Mycobacterium tuberculosis is the causative agent of TB. Gonorrhea is called Neisseria gonorrhea. And anthrax is also a livestock disease caused by a bacteria. Let's proceed to question number 3. During a microscopy practical, the following materials were provided: a temporary mount of an onion epidermis, a transparent ruler. State the aim of the experiment. State the aim of the experiment. So we already have a temporary mount of an onion epidermis on a microscope, that is, and we have a transparent ruler. So the aim of the experiment is to estimate, the size of a cell. It is to estimate the size of a cell. It is to estimate the size of a cell. To estimate the size of a cell. The next question reads, explain how the aim stated in 3A can be achieved. How can we be able to achieve that? The way we can be able to achieve it is, the first thing we are going to do is we will use the ruler to determine, use the ruler to determine the diameter of the field of view. The diameter of the field of view. The diameter of the field of view. Then the second one is that we will count the number of cells. We will count the number of cells we will count in the number of cells across the field of view. Why are we doing this? Because we want to determine the size of one cell. So we will count the number of cells across the field of view. Step number 1 was determining the diameter of the field of view. The second one is counting the number of cells. Then the next one is we will calculate the size of cell. So we will calculate the size of cell. And how do we calculate the size of cell? The formula is diameter of the field of view over the number of cells. Over the number of cells. And by doing so, we will have determined the size of a cell. So in that question, you will be awarded 3 marks. You start using the ruler to determine the diameter of the field of view. The second part is you count the number of cells across the field of view. And the third one is you calculate the size of cell. And how do we calculate the size of cell? It is the diameter of the field of view divided by the number of cells. The number of cells. Let us proceed. The next question, that is question number 4. The following diagram represents a specialized animal cell. We have a specialized animal cell. And what animal cell is this? This animal cell is referred to as a sperm cell. It is referred to us as a sperm cell. It is a sperm cell. It is a sperm cell. So this one is a sperm cell. So it is a sperm cell. So you can call it a sperm cell. It is a sperm. Or you can call it as a spermatozoa. But let us just stick with the sperm. The second question, name the cell organelle that is likely to be abundant in abundance in the part labeled F. So the part labeled F is referred to as the middle piece. This part labeled F is referred to as the middle piece. And this middle piece mainly consists of what we call the mitochondria. And because they have said in biology you must be very, very specific, we have been told about the cell organelle. So name the cell organelle, it is the mitochondrion. And what is the role of the mitochondrion in fertilization? The role of the mitochondrion, plural mitochondria, is to generate energy needed by the sperm cell to propel. So for propulsion, energy is needed, and that is why we have numerous mitochondria within the middle piece region of a spermatozoa. We proceed to the next question. Explain the answer in 4b above. I think we've already explained it. So what we now need to say is that the mitochondria enable the cell to respire, hence provide energy. It enables the cell to respire, hence provide energy. To respire, hence provide energy. Hence provide energy, which aids in propulsion. Which aid in propulsion. Which aid in propulsion. Number 5. Name 2 components of blood that are absent in tissue fluid. So the 2 components of blood that are absent in tissue fluid, number 1, we have the red blood cells. We have the red blood cells. They are also called erythrocytes. We have the white blood cells. Which we call the leukocytes. So what happens in this scenario? The tissue fluid is the fluid that is filtered out from the blood, and this fluid, it contains some nutrients such as glucose, the amino acids, but you find that larger blood proteins such as albumin and fibrinogen are not filtered out because they are large molecules and cannot be able to fit in the intracellular air spaces or the intracellular spaces. So we have the red blood cells, the white blood cells, and the proteins. Those are, to be specific, the blood proteins. These proteins cannot be able to pass through the intracellular spaces due to their large size. due to their large size. Due to their large size. Let us continue to the next question. So the next question is, name the structure in which plants through which, name the structures in plants through which the processes of transpiration and guttation occur. Good question. The process of transpiration and guttation occurs. So transpiration is the process by which plants lose excess water in form of water vapor. And the structures in plants where transpiration takes place is the stomata. The structures where guttation takes place, we call them hydrathodes. You may be asking yourself, what are hydrathodes? Hydrathodes are structures found in temperate plants. They are found in the leaves of temperate plants. Temperate plants are those plants that grow in extremely cold and permanently frozen places like Norway. So you find out that their leaves have got openings at the margin, and these openings lose water in form of water droplets. These openings are what we call hydrathodes. These openings are what we call hydrathodes. Hydrathodes. They are known as hydrathodes. Known as hydrathodes. We continue. Number 7. Name 2 classes of the phylum Arthropoda that have acephalothorax. Good question. Name 2 classes of the phylum Arthropoda that has acephalothorax. So the 2 classes of phylum Arthropoda that has acephalothorax, number 1 is the class Kcse. It is the class Kcse. And the class Arachnida. So listen and listen carefully. You must be very, very careful when it comes to spelling mistakes. Ensure you don't make any spelling mistakes. Class Kcse and class Arachnida. So this is found in the topic classification 2, form 3. First topic, classification 2, form 3, topic 1. Ili uende usome hizo. We have got phylum Arthropoda and phylum Chordata. And we have, for phylum Arthropoda, we have class Kcse, we have class Arachnida, we have class Dipropoda, we have class Chiropoda, and we have class Insecta. Those are the different classes within the phylum Arthropoda. You can go and also study in depth concerning their characteristics. Name the source of hydrochloric acid in the human alimentary canal. So where does this hydrochloric acid come from? The hydrochloric acid comes from the gastric glands. It comes from the gastric glands. So it is the gastric glands that releases the hydrochloric acid. And to be specific, in the gastric glands, hydrochloric acid is secreted from the oxynitic cells. From what we call the oxynitic cells. But just by saying gastric glands, you are good to go. Kisema tu gastric glands, you are good to go. If you have a question, post your question on our comment section. This is Top Grade Academy, where we turn confusion into clarity. If you have not subscribed to our YouTube channel, turn on the notification bell so that each and everything that we are uploading, each and every content that we are going to be uploading, each and every live streaming, you will always be updated. So do us a favor by subscribing. If you are in need also of our resources, you can access our website at www.topgradeacademy.co.ke, where you can get our free past papers with their specific marking schemes. So let us now go to B. 8B. The following diagram shows a process along the mammalian digestive system. Name the process. So what is this process called? This process, you can see there is a food bolus. This process is called peristalsis. Peristalsis, and it takes place within the esophagus region. So it is called peristalsis. Peristalsis. And it occurs during the process of swallowing. Yeah. State two roles of the process in digestion. So what is the function of this process during the digestion? The roles of this process called peristalsis in the process of digestion. Number one, it facilitates the movement of food. It facilitates the movement of food. That is number one. Another one, it also enables mixing of food. So, why are we talking of mixing of food within the alimentary canal? Peristalsis does not only occur within the esophagus, it even occurs in the intestines. So, what it does also, it also helps in absorption of water within the large intestines. It also helps in absorption of water from undigested food that is in the large intestines. In the large intestines. That is it. Let us continue. So this is very, very crucial. This is in the topic nutrition form 1. And there are so many essay questions assessed from this topic. So please ensure you learn the process of digestion. Number 9. Name one blood disorder caused by gene mutation. Name one blood disorder caused by gene mutation. So one blood disorder caused by gene mutation, we can give an example of sickle cell anemia. This is a question in Form 4 that is in the topic genetics. Sickle cell anemia or anemia. So this one is in the topic genetics. Form 4s in the room, ensure you study this genetics. Let us continue. Tuendelee, tusonge tukisonga. That is in the topic genetics. The next question, number 10. Name the stage in meiosis where each of the following processes occur. Formation of spindle fibers. So spindle fibers in meiosis are formed in the stage metaphase 1. Spindle fibers are formed at metaphase 1. Metaphase 1 is where the spindle fibers are formed. Metaphase 1. The second one, disappearance of the nucleus. The disappearance of the nucleus. So the nucleus disappears from the stage of meiosis prophase 1. Prophase 1. Remember, we are dealing with meiosis, so it is prophase 1. It is prophase 1. This one is in the topic reproduction. It is in the topic reproduction and cell division. Under cell division. Let us embark on number 11. Number 11, the following diagram represents part of the human male reproductive system. So we have the male reproductive system. We have part H, we have part G, and we have part J. So the question reads, name the part labeled G. Name the part labeled G. So the part labeled G is known as the prostate gland. It is known as the prostate gland. The prostate gland. It is known as the prostate gland. The second question, state one function of the structure labeled H. Structure labeled H. So tunafaa tujiulize, what is H? What is H? What is H? So G is the prostate gland. We now have H. H. So the function of H is known as the seminal vesicles, and the function of the seminal vesicles is to provide an alkaline fluid provides an alkaline fluid to neutralize the acidity of the vagina. To neutralize the acidity of the vaginal fluids. To neutralize the acidity of the vaginal fluids. That is one function. And another one is also nourishment of the spermatozoa. So you can write any of them. Nourishment of the sperms. Nourishment of the sperms. Nourishment of the sperms. The nourishment of the sperms. Hawa ni kina nani wako class na hawaongei? Are you understanding? Tunaelewa? Or we are lost along the way? Are we understanding or are we lost along the way? Tunaelewa ama tunapotea? Haya. Haya, tuendelee. Tuko pamoja? Just a minute. So we continue. The next question reads, how do the following structural modifications in plants minimize the rate of water loss? How do they minimize the rate of water loss? Number one, leaf folding. So what does leaf folding do? Leaf folding, what it does is that it reduces the surface area exposed. It reduces the surface area exposed. The surface area exposed. It reduces the surface area exposed. Sunken stomata. Sunken stomata, what they do is that it reduces the diffusion gradient hello, inside and outside the leaf. Number 13, state 2 reasons for the absence of complex excretory organs in plants. Why are there fewer or absence of complex excretory organs in plants? Number one is that some waste materials are recycled or reutilized. Some waste materials are recycled stroke reutilized. So one thing first I want you to understand is that, for example, plants use or utilize carbon dioxide for the purpose of photosynthesis. They give out oxygen. The oxygen given out as a byproduct during the process of photosynthesis is reutilized by plants during the process of respiration. That is a good example. Another reason is because some waste are stored in plants tissues. Some waste are stored in plant tissues. Some are stored in plant tissues. Let us proceed to number 14. State the significance of each of the following characteristic in mammalian gaseous exchange structures and surfaces. Number 1, presence of rings of cartilage in the trachea. The presence of the rings of cartilage within the trachea. Number 1, it is to keep the trachea open. It is to keep the trachea open and prevent it from collapsing. Prevent it from collapsing. Number B, numerous blood capillaries lining the lungs. So the importance of the numerous blood capillaries is to maintain a steep concentration gradient. Maintain a steep concentration gradient. Number 15. What is the most appropriate method of estimating the population of black ants in a school playing field? This topic is in ecology. This is ecology, methods of population estimation. So we are being asked, what is the most appropriate method? Tunataka kujua the number of black ants mahali kwa uwanja wa shule. So black ants hatuwezi tumia capture. The best method to use is quadrat method. And we can also use capture. There is no harm. Those two methods ziko sawa. Question B. Why are shorter food chains advantageous in an ecosystem? Why are shorter food chains advantageous in an ecosystem? The reason why shorter food chains are advantageous within an ecosystem is because they have a fewer, or what we call less, trophic levels. They have fewer trophic levels. They have fewer trophic levels. And another one is because energy is conserved. energy is conserved. The next question, using an example, define convergent evolution. So this is in the topic evolution. The above question A and B was in ecology form 3. This one is in evolution. Evolution is the second topic in form 4 after genetics. Form 4 we have 4 topics: genetics, evolution, response, reception and coordination, and support and movement. So using an example, define convergent evolution. So convergent evolution is the modification of structures of organisms. It is the modification of structures of organisms of different ancestral origin to perform similar functions. An example is the wings of insects and birds. That is good. Number 16. Let's go to number 17. Explain how natural selection is advantageous to living organisms. Explain how natural selection is advantageous to living organisms. Natural selection. So, what happens with natural selection is the law or the theory brought forward by a scientist by the name Charles Darwin. And this theory stated or asserted that an organism is fit to survive within a certain niche if it has the adaptive features that will enable it to survive in that. If it lacks the adaptive capabilities of surviving in that niche, it is going to be selected out. And that is what brought forth what we call survival for the fittest, or this theory of natural selection that was brought forth by Charles Darwin. So what natural selection explains, or how is it of advantage in living organisms? It is because it serves to identify organisms with desirable traits. It serves to identify organisms with desirable traits that help them survive and reproduce. It also helps in eliminating organisms with harmful traits. Number 17. Name the blood vessel that carries oxygenated blood from the heart to the rest of the body tissues. From the heart to the rest of the body, oxygenated blood, that is aorta. Aorta is also considered the main artery. It is the main artery. It is the main artery. Tunaendelea vizuri. It is the main artery. The main artery. Number 18. Or 17. A Roman 2. State the role of the tricuspid valve in the mammalian heart. In the mammalian heart. So this being a valve, it prevents the backflow of blood. So it prevents backflow into the right atrium. It prevents backflow of blood into the right atrium. Into the right atrium or auricle. Into the right atrium. Into the right atrium. It prevents the backflow of blood into the right atrium. So we proceed to the next question. Why are people with blood group O referred to us as universal donor? So I hope we have universal donors in class. Personally, your teacher is a universal donor. Nyinyi sijui ama mko blood group O. I'm blood group O and I'm a universal donor. And I do donate blood. Huwa napeana damu. So I'm a universal donor. So the reason why people of blood group O are referred to us as universal donor is because blood group O lacks antigens. Blood group O lacks antigens. Blood group O lacks antigens. When we talk of antigens, we mean A and B. Hence no agglutination. Hence no agglutination. Hence no agglutination is the crumbling of the red blood cells of the recipient. The crumbling. Tuende number 10, number 18, sorry. Number 18, the following diagram shows germination in two different seedlings labeled L and M. Hata ionekani vizuri sana. Identify the type of germination shown in seedling L. Aha, nikiangalia M naelewa hiyo ni epigeo. Epigeo. L hiyo ni hypogeo. Hiyo ni mahindi. So the type of germination here is hypogeo. This is in growth and development. This one occurs in monocots. It occurs in monocots. Haya, tusonge tukisonga. Roman 2, give a reason for your answer in A above. So the reason is because the cotyledon remain below the soil surface. The cotyledon remains below. The cotyledon remains below the soil surface. The cotyledon remains below the soil surface. Wewe mnatoka mnaenda wapi? B. State one common function of the parts. State one common function of the parts labeled K in seedlings L and M. The part labeled K. K. So K ni part gani? K ni cotyledon. So it is for storage of food. The one common function is storage of food. Storage of food. Twende number 19. The following diagram represents a longitudinal section through a phloem tissue. So that is the phloem tissue. We have P and N. A. Account for the high concentration of mitochondria in the part labeled N. Account for the high concentration of mitochondria in the part labeled N. So part labeled N is the companion cells. The companion cells contain numerous mitochondria. So the reason is to synthesize energy. It is to synthesize energy for translocation. Translocation of food. The second one, it is needed for active transport. It is needed for active transport. And the other one is, this is the active transport. It's just a continuation. Active transport of food substances in and out of sieve tubes. The next question, state one structural adaptation of the part labeled P to its function. P ni gani? P. You can see the P. Yeah. Part labeled P. One structural adaptation of part P is that it is filled with fine cytoplasmic filaments. Filaments for streaming of food. For streaming of food from one sieve tube to another. Number 20. 20. 20. The following word equation represents a metabolic reaction taking place in an animal tissue. Glucose into becomes lactic acid plus energy. State the condition under which the reaction occurs. This one is anaerobic respiration, which occurs in the absence of oxygen. So the condition is it occurs in the absence of oxygen. The absence of oxygen. Part B. How does the size of an animal affect the rate of respiration? Number one is that small-bodied, you know that according to surface area to volume ratio, animals have large, I will short form it because of our screen, surface area to volume ratio that is for heat loss. And therefore, hence high rate of respiration. Hence higher rate of respiration. To compensate the lost heat. Haya, kidogo, just a minute. Haya, let us continue. What am I shortening? Nimeshorten nini? Mugule niambie. Niambieni saa hii iko aje? I will share this question paper. Nitawatumia hii question paper kwa group. Sawa? I will share it, don't worry. Nitashare question paper. Tuendelee. Hapo iko sawa? Number 21. How can sexual reproduction in organisms lead to the evolution of new species? So how can sexual reproduction in organisms lead to evolution of new species? Number 1 is it is because of gametes. So what you need to know is that gametes, they have different genes from different parents. So it is through gametes with different genes from different parents fuse to form new offsprings. They fuse to form new offsprings. They fuse to form new offsprings. So what happens is that the offsprings vary in characteristics. The offsprings vary in characteristics. They vary from their parents in characteristics. Part B. This is evolution. State the role of the continental drift in the evolution of organisms. The role of the continental drift. So this is a proof that evolution took place. And the role of continental drift in evolution is that it led to separation of organisms. It led to separation of organisms. And these organisms, they were separated from their initial origin or from the same origin to different environmental conditions. To different environmental conditions. To different environmental conditions. Hence acquiring adaptive characteristics. Number 22, that is also evolution. Number 22, the following diagram represents an experimental setup used to investigate a certain biological process. We have oil, and we have boiled and cooled glucose plus yeast suspension. Then on the other end, we have lime water. So the first question reads, identify the biological process that can be investigated using this setup. So the process here is anaerobic respiration. Anaerobic. Because oil is there, so it takes place in the absence of oxygen, which we can also call fermentation. Which we can also call fermentation. The second question, give a reason for the answer in 22A above. The reason is because boiling removes air. The reason is because boiling removes air. And the layer of oil and oil prevents the entry of air. It prevents the entry of air. That is why it is anaerobic respiration. Write a word equation. So you can see there was glucose and yeast. So what is happening is that glucose in the presence of yeast will form ethanol plus carbon dioxide plus energy, which is ATP energy. So that is the equation of anaerobic respiration in plants. Glucose, ethanol plus carbon dioxide plus energy. Part C, suggest a modification on the setup that would increase the rate of reaction in the conical flask. So how can we be able to increase the reaction within the conical flask? Number one, we can increase the temperature. When we increase the temperature to optimum, that will increase the rate of the reaction. Number two, the second thing we can do, we can increase the concentration of yeast. When we increase the concentration of yeast, more energy will be formed because there will be an increase in concentration or an increase in rate of anaerobic respiration or fermentation. Let us go to part D. Why is it necessary to cool glucose? Why is it necessary to cool glucose before adding yeast in the conical flask? So the reason why it is important to cool glucose before adding yeast is to avoid killing the yeast cells. It is to avoid killing the yeast cells. To avoid killing the yeast cells. That is the reason, or denaturing the enzymes. of the yeast cells. Tunasonga. The next question is number 23. The following diagram represents a bone obtained from a mammalian skeleton. Hii bone ni gani? Hata nyinyi mkiangalia mnaona hii bone ni gani? Hiyo bone mnaona ni gani? Nipatieni jibu hapo kwa comment. Hiyo bone ni gani? What kind of a bone? That is number 1. It is part of the cervical vertebrae. It is part of the cervical vertebrae, the neck region. But within that neck region, which bone is it to be specific? Hiyo bone ni gani? Jibuni hapo. Nataka kuona jibu saa hii. Which bone is it? Which bone is it? Axis, karibu sana. Iko sawa? It is axis. It is an axis. Correct. So an axis is a bone within the cervical vertebrae, and how do you know it is an axis? Umejuaje ni axis? The reason why it is an axis is because of the structure R, which we call the odontoid process. Ukiona tu hiyo mshale inakaa hivyo, wewe ujue hiyo ni odontoid process. Odontoid process. Cha tuandike odontoid. Odontoid process. Tuende part B. Name the bone that articulates at the point S. Ni bone gani ina articulate at point S? Hapo S juu inakuanga atlas. And atlas has a very huge centrum. Then yenye inashikia hapo chini ni other cervical vertebrae. The other cervical vertebrae. Why do we say other? Juu si moja. There are 5 of them. Other cervical vertebrae. Other cervical vertebrae. In total, the cervical vertebrae has 7 bones. The first one is the atlas, the second one is the axis, the rest are 5. So we have the third, the fourth, the fifth, the sixth, the seventh. So yenye kumeshikana hapo si ya tatu. The third cervical vertebra. Tuende the next question. Number 24, ecology. Hiyo ni support and movement. The following diagram represents a bone. No, tushafanya hii. The following food web shows a feeding relationship formed in a certain ecosystem. We have green plants, they are eaten by antelopes and locusts. Locusts are eaten by lizards. Antelopes are being eaten by cheetah and lions. Locust is being eaten by lizards. Locusts are also being fed on by frogs. Frogs are preyed by hawks and snakes. Snake nazo zinapreyiwa na hawks. Good, that's a food web. From the food web, identify the organism with the lowest biomass. Which one has the lowest biomass? The one with the lowest biomass is the top notch when it comes to the feeding level or the trophic level. So the one with the lowest biomass should be the hawks. Judy top on the trophic levels. Then trophic level, identify the trophic level occupied by lizards. Lizards occupy in producers, primary consumers, secondary. So lizards occupy secondary consumers. Name the type of feeding relationship between the lion and the cheetah. Lion and cheetah zote mbili ukiangalia zinakula antelope. So what kind of a feeding relationship do they have? Competition. They compete. So the feeding relationship is competition. Cheetah and the antelopes is predation. Predation. Part C. Explain the role of bacteria. Explain the role bacteria would play in this ecosystem. So the role bacteria would play in this ecosystem would be, the role of bacteria is decomposition. It is decomposition. And what is the purpose of decomposition? For releasing nutrients in the ecosystem. For releasing nutrients in the ecosystem. We are done. How was the class? Imekuwaje? Umeipenda? Kama umeipenda, like. So if you are in need of materials, check out from our website www.topgradeacademy.co.ke. If you are in need of tuition enquiries, the number is +254 742 09 08 25. So our class is over. We are done. Tumefanya hiyo. Tumemaliza hiyo ya leo. Tomorrow We'll be checking out on mathematics paper 1. Mathematics paper 1 2024. Mathematics paper 1 2024. Class imekuwa poa. So we've come to the end of our session.